hosting this webinar. So with that as a backdrop, why don't we begin? The presenters today will be myself, again from the Division of Water. I'm going to take a little bit of time to give you an introduction to Hydrilla, give you a little bit of a basis for understanding where the plant is, something about the plant, and then I'm going to hand that off to Bob Johnson from Racing Johnson Aquatic Ecologist, who is going to give you some good inside information about how hydrilla can be identified, because as you'll learn, there are a number of aquatic plant species in New York State, both native and exotic, that look a lot like hydrilla. Then Bob is going to hand it off to Greg Sargis from the Nature Conservancy out in Western New York, and he's going to talk about how to survey for hydrilla and go over a number of the survey methodologies, including a brief discussion anyway of the work that he's doing as part of a project with the Nature Conservancy. Greg is going to hand it off to Meg Wilkinson from the Natural Heritage Program, who's going to talk about IMAP and mechanisms for reporting this information. And you'll hear this several times during the presentation, but we want to know when you've looked and where you've looked, even if you haven't found hydrilla. And again, Meg will talk about that in more detail. Meg will bring it back to me. I'll close the webinar, and then we'll open it up to more formal discussion. But again, if you do have questions during the process, don't hesitate to click the chat button and let us know what kind of questions you're having. So we're going to spend some time today asking you to help us. So the first part in the process is learning more about hydrilla. And for those who are already familiar with the plant, I'll apologize for what may be some redundant material. But for those who don't know much about hydrilla, we hope you'll learn something about it today. Then we really want you to help us find hydrilla. Look for it, collect any suspicious plants, document what you're looking for through photographs and collection of plants, report your findings, ideally through IMAP, but it doesn't necessarily have to be through IMAP. And then we'd like you to do this preferably over the next 2-week period of time. We'll talk a little bit about why this particular window is important, but we do want to emphasize that any time you're out looking, whether it's later on this year or next year, if you see something that looks like hydrilla, remember what you learned today and how you can report that information to us. We also hope that you'll take an active role in working with us and your local PRISM, which is your Partnership for Regional Invasive Species Management, to help eradicate any populations that you find. First and foremost, we hope you don't find any, but if you do find it, we hope we're going to find it early enough that we can do something about it. And finally, we hope you can take this information and help to educate other people about hydrilla. So the outline today, again, is we're going to talk a little bit about hydrilla, why you should care about it, a little information about the biology of hydrilla and where we've seen it in New York State. Then Bob is going to talk about identifying hydrilla and distinguishing it from common lookalikes. Then Greg is going to talk about how to survey for hydrilla. Meg is going to talk about how to document, record, and confirm findings. And then we'll come back to, again, what your role is in the process, what the next steps are, and some places where you can find some additional information. So before we talk about hydrilla, we should probably have a brief discussion about invasive species, and very specifically aquatic invasive species. And there are really 4 take-home messages. Now, we could have an entire webinar talking just about aquatic invasive species in a general sense. There's a lot of important information that we're really not going to convey here, but we do want you to have a couple of take-home points here. The first is that invasive species, aquatic and terrestrial, are both non-native and destructive. And those 2 terms are important. We characterize invasive species as exotic species, those that are accidentally or purposefully introduced outside of the historic range. Then the destructive element is also important, that these are organisms that rapidly reproduce and can displace native species, resulting in ecological and economic impact. So both parts of that definition are particularly important. The second take-home point is that aquatic invasive species are prevalent throughout New York State. And as you can see, there are both aquatic plants and aquatic animals, and a large number of them that are at the very least exotic and in certain circumstances may be invasive. Many of these species are generalists, and they can survive in a wide variety of environments. And aquatic invasive species that are the most problematic are the ones that can spread and reproduce through multiple reproductive strategies, and they can ultimately, in the process of doing that, in the process of creating their explosive growth crowd out both native and other exotic species. So with those as sort of the four key take-home points, how does hydrilla fit into that mix? Well, hydrilla is a non-native and destructive species. It's not native to North America, the United States, or New York State, so it is not native. It can be destructive. It can produce very dense mats on the surface and throughout the lake. New York State has gone through a preliminary process of doing an assessment of invasive species, and at the time, hydrilla was identified as highly invasive, which is consistent with this federal definition of the lake as a federally noxious weed. So again, that first take-home point, non-native and destructive, invasive, is important, and hydrilla fits the bill. The second point is that these are species that are prevalent in New York State. Hydrilla was first found in Orange County and is now found in a number of different locations. So it's found in a bunch of different places. The third point, many AIS are generalists that can do well in a wide variety of environments. And you can see from the slide there are a wide variety of habitats in which hydrilla does very well, not only in other parts of the country, but here in New York State where we found hydrilla. We found it in shallow and deep water. We found it in a wide variety of sediment types. We found it in a wide variety of water quality characteristics. And we found it essentially every kind of place that you can find aquatic plants to grow. That gives it a tremendous advantage over native plants and even some other exotic plants. So that's one of the reasons it does extremely well. And AIS can spread through multiple means. As you'll learn in a few minutes, it has a number of different ways that it can reproduce, both from tubers and from turion. So I mentioned this briefly, but I'm showing the map in a little more detail. We found this in close to a dozen different locations in New York State. The original infestation was found in Orange County in 2008 in a relatively small pond that had a mucky bottom. Then it was found in the same year in Long Island, and in the last few years we found troublesome populations and quite extensively growing populations in Cayuga Inlet and the Erie Canal. And then the last infestation found last fall was in a few ponds in Broome County. So in the discussion about aquatic invasive species generally and hydrilla specifically, we indicated the kind of problems that an ecological environment and an economic environment, if you will, are faced with in the introduction of hydrilla. But I want to emphasize a couple of other key points. Why we care about this, one illustration of that is to look back, turn back the clock, if you will, to the 1940s. The map on the upper right part of the screen shows the distribution of Eurasian water milfoil as of 2012. If you had looked at a similar distribution map in the 1940s, you would have seen at most one or two dots in the Finger Lakes region, the area where Eurasian water milfoil was introduced. At that time, and really over the intervening 20 or 30 years, there weren't a lot of tools available for eradicating populations. And frankly, we didn't know a lot about invasive species and didn't know how important it was to try to eradicate these populations. So now if you look at the infestation of hydrilla, as noted in the previous slide, it's been found in a number of places in New York State. It's been found in Long Island, it's been found in western New York, and a number of points in between. The most recent infestations show that it is now at the portal of the Great Lakes Basin. It is perched to go right into the Great Lakes, and it would be a new explosive invader in many of these water bodies. So if you look at the bottom right part of the screen, it shows a plot. Many of you have seen this already, and let me just take you briefly through this. This curve shows the change in the abundance of an invasive species over time. And if you look at the bottom part of this graphic where you see the plant is initially introduced, there is obviously a small introduction. Most of these invasive plants are introduced a single plant at a time. So the abundance is very low. And it stays that way for a period of time until ultimately it starts to colonize into beds and starts to form a more significant growth pattern in a water body. So we move further up the curve to the point where it is first recognized by people. And really it is only recognized even early in this plot as it's starting to move up this distribution scale when there is active vigilant searching. As you get further up the graph, you'll see that the abundance increases substantially, and then as the abundance increases, that's when public awareness typically increases. Apologize for the moment. We're having a small technical difficulty that we hope will be resolved in a second. Okay, sorry about that. When you get up to the top of the curve, or near the top of the curve, is when public awareness typically kicks in. At that point, the abundance is significant, and both the ability to control these invasives and the cost associated with that control can be so substantial that effectively you can no longer eradicate that population. Getting back to where we started with this, Hydrilla is essentially now where Eurasian milfoil was in the 1940s, and if we could turn back the clock, we would devote a significant amount of time and attention to try to eradicate these populations. That's where we are now. So, why we're here today is that we need your help. As noted in the previous slide, the only real chance we have of eradicating Hydrilla is to keep both the infested number of water bodies small and to catch the infestation early enough in its infestation cycle within a water body that you have a real chance of trying to eradicate that population. That's where we think we are with most of the infestations that have been found in New York. So as a result, we're devoting a significant amount of time and effort and resources to try to eradicate these populations. So we need your help to try to find these infestations early. And it's not going to be easy once we find them. There are a number of challenges, and I'm not going to go through all of these points, but the key ones are that this is a plant that has two different biotypes, and again, we're not going to go into the details, but there's a monoecious variety and a dioecious variety. The monoecious variety, the one that we have in New York State, we know very little about. Almost all of the research in this country, more than 90% of the research papers on Hydrilla have been in association with the other variety. And those who have been studying this plant for many years believe that these are effectively two completely different plants. So that's a challenge. Another challenge is that it's been found in a wide variety of water bodies, including some flowing systems that are particularly challenging to try to control those populations. There are limited resources that are available. New York has no statutory authority to intervene as a general rule in all of these infestations. So we try to find opportunities where we have the ability to intervene and do that intervening. And the last point is that Hydrilla can be very difficult to control because it's difficult to find. And it's difficult to find because it looks so much like both some common plants in New York State and some exotic plants in New York State. So, with that, take a look at this picture, and in your mind, tell me what you think it is. I guess you can't really tell me in your mind what you think it is, but look to see what, look at this plant and look at the kinds of plants that typically look like this and see if you can identify what this plant is.